I'm HyTest Russia CEO Yulia Belausova. Welcome to HyTest Russia.
Hello, I'm Tatiana Yasakova, Production Director of HyTest. I will show you our production premises in HyTest Russia. Here we produce all our well-known antigens and antibodies, more than 500 totally in the HyTest catalog. We have few production groups here in production that are special functions, each one. We have Animal House, where we produce aesthetic fluid for our in vivo antibodies. We have Cell Bank Group that supplies all needed cell material for our work. We have In Vitro Group that deals with all raw materials for in vitro and recombinant antibodies as well as recombinant antigens production. Then we have group dealing with antigens who purify them and make the quality control primary testing, and we have antibodies group that purifies all our types of antibodies and do quality control test as well. So let's see what we have here. Let's talk about one of the key things of HyTest products manufacturing, our cell banks. We have 3 places of the storage for safety reasons, and one of them is here. This room has limited access. Only certain persons can enter who works with the cells, and here we store our master cell banks as well as working cell banks used for antibodies manufacturing. The work itself with the cells is done in the laboratory that you see now. Here we test the cells quality and prepare them for inoculation into mice or into bioreactors. Our traditional product group is antibodies produced in aesthetic fluid or in vivo. For this purpose, we don't breed mice by ourselves. We purchase them from certified suppliers. And we conduct aesthetic fluid production during more or less 1 month per each production cycle. Here you can see the conditions where our mice live and the room for manipulations with them. We are now in the clean rooms of our in vitro production group. Here all the materials are prepared for in vitro antibodies, recombinant antibodies and recombinant antigens further purification. For in vitro produced antibodies we use bioreactor of New Brunswick type and perfusion mode of production that allows us to get 20-25 grams of the product through one cycle. Also, we conduct small-scale production in these CO2 incubators in flask-type bioreactors, cell line 1000. We also work with different types of the cells, for example, insect cells used for production of some recombinant antigens. And the equipment that is used for this purpose is another type of shakers. The antigens production starts from the raw material. For native antigens it's tissues, for recombinant antigens it's cultural fluid produced in our in vitro group. Then every antigen is purified according to a specific protocol. And to do it we have the whole set of all needed equipment for those purposes. We use different set of chromatography processes and we conduct them on automated systems as well as normal pressure systems. Also we use different equipment for centrifugation and a lot of specific equipment for quality control tests, such as image quant scanner, vector fluorimeter, and others that are typical for such purposes. Another big production group is antibodies group. Here we purify our antibodies from acidic fluid produced in the animal house or from cultural fluid obtained in vitro production group. The purification procedure itself is classical. We use protein A purification practically for all our antibodies. And chromatography systems that we use are of normal pressure as well as high pressure machines. You can see the process here in this room. Also, we conduct primary quality control tests for all our products that then are accepted in our quality control management group. So here we have all the equipment needed for this purpose, testing of immunological activity of antibodies, their purity and their concentration.
Administrative offices are located on the second floor. Shipment, HR, accounting team and also Russian sales are here.
Hello, this is HR team office. All employee-related documents, including employee training records files, are kept here under the lock and key.
So now you see that we have here in Moscow all needed capacities to ensure all production volumes that are needed for current level of the sales. And moreover, when we move to new premises in Nagatina, we will receive a new space for the further growth of at least a few years.
Dear colleagues, my name is Alexey Katrucha, and for many years I am working as R&D director of HyTest. And today it's my big pleasure to be your guide on our short tour of HyTest R&D new premises. The first place that we will visit during our tour is the premises that belong to a group of gene engineers. Usually any project devoted to the antibody development starts from the development of recombinant proteins, which are used as antigens for animal immunization or standards and calibrators in our immunoassay prototypes. Production of recombinant proteins in bacterial cells is an essential step of many projects focused on recombinant protein development, since bacterial cells are recognized robust and powerful protein expression systems. In this room, we select and propagate individual bacterial clones for either subsequent DNA isolation or for induction of protein expression. Quite many proteins were producing in the insect cells, or more technically with baculovirus expression systems. This is the combination of insect cells and recombinant viruses. Recombinant human thyroperoxidase and several veterinary products were obtained from insect cells infected with specially designed baculoviruses. For the development of glycosylated antigens and recombinant antibodies, we are using mammalian cell lines such as HEC293 and CHO cells. For this purpose, our gene engineers use different equipment that helps significantly shorten the development time and improve the quality of our products. Flow cytometry is a technique used to detect and to measure physical and chemical characteristics of population of mammalian and bacterial cells and is used in the cell line development process. EVOS fluid fluorescent microscope captures high-quality fluorescent cells images right at the benchtop within a minute. This microscope is used by our specialists for the analysis of transfection efficiency, retroviral infection process, and cell stability assays. On the next step, obtained recombinant proteins are purified and analyzed in our biochemical laboratories. Currently, we have 5 biochemical laboratories similar in organization. Protein purification is carried out using normal pressure and FPLC chromatography systems. Currently, we have several FPLC systems which are used for protein purification and for quality control procedures. The protein purity is analyzed by electrophoresis. The detection and analysis of the proteins after electrophoresis and after Western blotting is performed by means of ChemiDoc Touch Imager. This is a modern detection system, enables fast and sensitive imaging of proteins, including fluorescent and chemiluminescent detection, as well as further quantification of analyte. Obtained proteins are used for animal immunization and later on for the development of monoclonal antibodies. Currently, we are developing mouse, rabbit and rat antibodies, and in our nearest plants, antibodies from pigs and from guinea pigs. In these premises, we are working on obtaining hybridomas, the future source of our monoclonal and different forms of recombinant antibodies. On the first stage of hybridoma development, we have a lot of work related to antibody testing. And we have significantly simplified this stage by using Tican robot station. That is octet device used for label-free real-time affinity measurements. We use it for early screening of hybridoma clones and also to measure affinity constants of interaction between purified antibody and the antigen. Switching from hybridoma-produced antibodies to recombinant ones demands knowing the exact sequence of variable regions of heavy and light chains of antibodies. There are several sequential steps including RNA acylation, reverse transcription, PCR amplification of corresponding regions, subsequent sequencing and data analysis. For the first time we have implemented this approach for mouse hybridomas. Now we're working with rat and rabbit antibodies. On the last stage of antibody development, we're analyzing obtained antibodies in sandwich immunoassays with purified antigens and later on with patient samples. We are releasing only those antibodies that could be used for the development of the immunoassays recognizing and light in the patient sample. As you have seen, new R&D facilities is a great place to work, and we're looking forward for the time when we can join with our production and office teams.